I'm Nick Patterson. I'm a mathematician by training, but I've had multiple careers. I've worked in cryptography for the US and British governments. I worked in finance, modeling financial markets, and now I do genetics. And those look like three very different disciplines, but all of them involve mathematical modeling of complicated data. And that's what I think I've learned a little bit about how to do. You do learn a common skill set. The actual material in cryptography, in the financial markets, in genetics, obviously completely different. And you need to learn enough about the underlying disciplines to make sure that what you're doing makes sense. But the modeling definitely has things in common, and some of the technical techniques I use map across directly. I also think that you learn a skill set about is this model working on this data? And that's a kind of fuzzy question, and I think I've been working technically for a very long time now, and over the years you acquire fairly deep expertise in how you can answer that. I have what I call my smell test, which is, does this data look right? And that can be really quite useful. But today we're going to talk about genetics, and in particular using genetics to learn about human history. So what happens in genetics, as of course you know, you get your genome, your genetic material, from your mother and your father. And the genetics, the genome involves strings of information in a 4-letter alphabet. So for example, humans have 2 chromosome 3s, which you can think of as strings of A, C's, G's, and T's, that's 4 chemicals. And you get one of your chromosome 3s from your mother and one of them from your father. Well, what happens if you look back at your further ancestors? Well, about a quarter of your material comes from your father's father. And if you go back 10 generations, you have a lot of ancestors, and the genome is being contributed to by multiple ancestors. That's just a little bit of background. What happens is the genetic variation starts to differ between populations that aren't exchanging genetic material or aren't mating with each other. So over thousands of years, if 2 populations separate, the genomes start looking very, very slightly different. So for example, it's quite easy to tell genetically if someone comes from West Africa or comes from Europe. You may make very, very occasional mistakes because you may have an African immigrant into Europe or something like that. But by and large, it's pretty easy to do that. And we can do much subtler things. If you tell me, here's half a dozen people, and they're either English or Italian, make a guess, I will get that right nearly every time, because there's just been enough separation between the history of England and the history of Italy to make that technically possible. So we're all walking around with genetic material from our ancestors in the distant past. I like to say we're walking around with our history. And what I'm trying to do in general is take human DNA and learn about that history. So we were doing that 10 years ago with some success using modern human DNA. But now there's been a massive technology shift. It's now possible to recover genetic material from ancient bones. And this changes everything, because instead of having to try and deduce mathematically what the ancient material must have looked like, we can now go and look directly. It's much, much easier. So let me say a little bit about what's now technically possible. You take an ancient fossil and you try and recover DNA from that. Actually, even with the best modern methods, more than half the time you fail. The DNA degrades in the ground, other bad things happen, bugs eat it up, and there's no certainty at all that we'll actually get useful material. But some of the time we do. And it can be very, very old. One of the first major successes, which I was involved with, working with a group in Germany, was recovering a DNA sequence of a Neanderthal who died in Siberia more than 40,000 years ago. And the DNA we recovered was of the same quality as I would get from taking a blood sample from you and running it out in a top quality lab like we have here in the Broad Institute. So 40,000-year-old DNA is essentially fully recoverable. So how far back can you go? Well, we're still working on that. The record on the human lineage is DNA recovered from a material in Spain from about 450,000 years ago. The DNA is not very good quality, but we got a reasonable amount. And the record for any mammal, not done with my group, is a fossil horse buried in northern Canada 750,000 years ago, where the DNA quality was superb. The conditions of burial were absolutely ideal. So that's some technical background about what we can do. What kind of materials do we need to extract DNA from? Bones, but we've actually learned quite a lot that not all bones are created equal. Somewhat to my surprise, big bones like the big leg bones are not very good. What happens after death, it seems, is that bacteria get into the bone and essentially eat up all the DNA we're interested in. So the big leg bone's not so good. Teeth are not too bad. And a discovery has been made just in the last couple of years for a bone called the petrous bone, which is involved in the ear, behind here. And this is very, very hard. And it seems that it's so hard that the bacteria are not able to get in. So we've had fantastic success with these particular bones from the skull. And that's been another technical advance, which has greatly improved the probability that we actually successfully get material. So I and my co-workers are really interested in all of human history, and we are interested in collecting ancient material really from anywhere in the planet. We of course have to rely on collaborators, archaeologists in particular, who've done excavations and often find human remains as part of the excavation. So, for example, we have a remarkable set of fossils from ancient South America, 8,000-year-old bones from the Andes in Peru, who, very interestingly to me, look remarkably genetically like modern Quechua Indians in Peru. There's clearly a quite straight line of descent there. We have material from Asia, all over Asia, ancient Iran, and especially in Europe. The density of material in Europe is much higher than elsewhere, simply because the archaeologists have been working harder in Europe than elsewhere on the planet. So we've been looking especially at ancient history in Europe, and let me tell you a little bit about that. So my own opinion is that this ancient DNA work is less interesting, though not completely without point, when we get nearer modern times and the historical societies are literate, so they are writing. The point is, in a society which is writing, there's typically very deep evidence about what happened simply from texts. So if you look in ancient Greece or Rome or ancient Mesopotamia, where there are enormous amounts of material, we already have a pretty good idea of the history. But if we go back further, everything's much less clear. So in Europe, we now have a really very interesting story, and I'll take a few minutes to tell you about that. So let's go back to about 8,000 BC in Europe. At that time, there is no farming going on in Europe. There's farming going on in the Middle East. And the people living in Europe are hunter-gatherers. So they are hunting animals, big game, and they are collecting vegetables and roots. And we think they're semi-nomadic because you have to follow the animals and so on. And the first discovery we made is that the genetics across Europe are remarkably homogeneous. So you look at people from ancient Spain, 8,000 BC, or in Germany or Hungary, genetically they're really quite similar. But if you go further east onto Finland or the Russian steppe, you see a big genetic shift. We're still thinking about exactly why that happened. But these hunter-gatherers do not look genetically at all like modern Europeans. The next thing that happens is farming arrived in Europe. And there'd been a question for a long time about whether farming came in because the old hunter-gatherers learnt, probably from the Middle East, how to do it, or did new people come into Europe? And we now know the answer. It's absolutely new people. And the proof of that is you get ancient DNA from very early farmers in Europe. It's completely different from the hunter-gatherers. Must be new people. And that's true. Farming arrives in Spain and Germany about the same time, a little earlier in Greece. All these early farmers look very similar genetically. But that's not the end of the story in Europe. The resulting populations still don't look like modern Europeans. And then something happens about 2500 BC, which is when the Bronze Age is just beginning in Europe. And what we now know is that there's a massive incursion from the Russian steppe. There's something like half or three-quarters of the genetics of the people at that time are not descended from first European farmers. They're descended from pastoralists who are grazing cattle on the steppes and come into Europe in a massive way. So we now have three sources of genetic information, of genetic material in Europe. We have the hunter-gatherers, we have the first farmers, and we have these steppe people. These three groups of people were all genetically very different from each other. And it turns out that modern Europeans are a three-way mix of those groups. None of this material could remotely have been discovered without doing ancient DNA. And it's completely changed how archaeologists are thinking about the ancient history of Europe. So one question is, we talked about these steppe people coming into Europe, and there was obviously a massive population replacement occurred. How did that happen? It's rather unlikely to have happened simply by peaceful negotiation. Until very recently, I thought it was almost certainly violence. The steppe people are riding horses, extremely useful in war, and I thought that they come in on horseback and the farmers can't really deal with that. But there's in fact another story which has become maybe even more likely. We're not sure yet. We've discovered that on the steppe as early as 3000 BC, the people there were carrying bubonic plague. And we think that in Europe, there was no plague at that time. That's a completely new discovery. The earliest the plague had been believed to occur was something like 500 AD in ancient Byzantium. But it's much, much older than that. So here's a story. Trading starts between the steppe and the farmers. That's very likely, and in fact there's archaeology suggesting that must have happened. And the steppe traders bring their little friends with them. And they've acquired, the steppe people have acquired some resistance to the plague, but the farmers are completely exposed. This is much as like what happened to first Americans when Europeans show up in the Americas. The Europeans are carrying lots of diseases like smallpox and measles, which is a complete killer if you have no resistance. And it devastated the first American population there. Perhaps something like that happened in Europe. Bubonic plague breaks out among the farming community. There's a massive population collapse. And there's then a kind of vacuum into which the steppe people can move in. We're still exploring that, but it's an interesting possibility and not violent in the same way as I originally thought. A personal interest of mine is how Indo-European spread. So linguists have divided up the languages of the world into very large families. And the largest family, both in numbers of people who speak it and the geographical spread, is Indo-European. And from the name, you might guess correctly that this is spoken by most of the peoples of Europe. Languages like French, German, English, Romanian, are all Indo-European, Latin and Greek as well, but also in India. Hindi, which is the majority language of North India, is Indo-European, and Hindi derives from ancient Sanskrit, which was being spoken in North India as early as 1500 BC. The spread geographically is even larger than you might know. There's a fascinating group called the Tocharians who were in Western China about 500 BC. They were literate. They wrote in Chinese characters so we can read their texts, and it's an Indo-European language. So by 500 BC, we know for certain that there are people speaking this language group on the Atlantic coast in Europe and also in Western China and also in North India. A very interesting and important question is, how did that happen? This question has been argued by archaeologists and linguistic theorists for nearly 200 years, but the DNA is now also giving evidence for this. We think that this steppe culture that came into Europe, almost for certain, were Indo-European speakers. They're a culture called by archaeologists the Yamnaya, and they flourished on the Russian steppe from a little earlier than 3000 BC to perhaps 2500 BC. They were the first peoples in the world to use wheeled vehicles, and probably they were also riding horses. So those steppe people are speaking Indo-European. There's an ancient population in modern Turkey called the Hittites that also spoke Indo-European, again from texts. We don't yet understand the genetics. The Tocharians I already mentioned, a real question. And my current research as of the last few months is to try and understand how Indo-Europeans reached India. And at the moment, we don't have any ancient DNA from India. We have wonderful collaborations, but India is a tough place to get DNA from because it's hot and wet. and this is bad for the preservation of genetic material. We do have ancient DNA from ancient Iran, and that's fascinating, and there's clearly a genetic relationship between ancient Iran and modern India, and I'm struggling to understand that in my own work. So what does my data actually look like? Well, it really begins as short pieces of DNA. And so let's back up. We have fossil material given to us by an archaeological collaborator, typically from an archaeological excavation. We make bone powder by drilling into the bone with an ordinary dentist drill, and DNA can then be dissolved from the bone powder and put into solution. You can then recover the sequence of 4-letter strings in the DNA. So at that point, we have a computer file which consists of short 4-letter strings. And what to do? Well, because this is human, that's the material we're interested in, it should strongly resemble modern human DNA. So we then take those strings that perhaps are 100 letters long, and we align them to modern human sequences. We don't expect to get an exact match, but you would expect that there'd be only 2 or 3 differences in 100 letters, and that would uniquely place the string on the human genome. So, backing up, we take the fossil, we make DNA in solution, we sequence it through standard modern sequencing machines, we align it to the human genome, and then we have ancient DNA aligned to modern human DNA, and we can look at what that ancient DNA looks like and whether the genetic variation is similar or different to other modern human populations. So how do we date the material? Well, the most common method is simply archaeologically. If you excavate a site, very often the archaeologists are extremely confident how old the site is. For example, there might be shards of pottery in the site, and they know enough about the pottery techniques to accurately date it. We also can do carbon dating, which is outside my expertise, but is standard now in the archaeology business. Finally, and this is really cutting edge, we have some methods which, in favorable circumstances, will actually allow us to date material directly from the genetics. It's very nice to do that because you get a cross-check on the material, particularly for the very old material. If it's 30 or 40,000 years old, the archaeological methods become very difficult to apply, and we now have some hope that we can do something with genetics. What I largely work on is recovering what's called phylogeny. So this is like a family tree, but at a population level. So you could imagine, for example, I told you about how Europe is a 3-way mix of populations. How do we know that? Well, first of all, we see the genetic shifts, and then there's ways of measuring how close a population is to another population. So I'm simplifying a little bit, but you see that the first farmers in Europe look very different from the genetic material we see on the Russian steppes. But by 2000 BC, we see people in Europe who clearly share a lot of genetic variation from the steppes, and we can model that mathematically, and the only reasonable explanation must be there's been some genetic exchange, mating, between the European peoples and the steppe peoples. Genetics is the area of biology which is most mathematical. And there's an interesting reason for this. It's that the basic processes of exchange of genetic material can be very simply described. How you get your DNA from your mother and your father, and how they got their DNA from their mother and their father. And there's a mathematical description of this, which is very simple and essentially accurate. The consequences of that are not so easy. But what mathematicians like is simple processes that produce complicated conclusions. And genetics is very much like that. So I build mathematical models, and so for instance, if I have an ancient population and a modern population, a natural question is, do I believe that this modern population could be directly descended from the ancient one, or has something else happened? And we have formal mathematical tests for this, many derived by other people in the community, some of them worked out by me. And so I take the DNA and apply these tests, and maybe it tells me, no, this population is not descended, something else must have happened. And then we try and think, how can we refine our models? So, for example, I have a formal test for whether one population is directly descended from another. And you take our data, you run that test, I decide, say, no, it can't be. And then we have to build a more complicated model to try and explain what we're seeing in the data. So in my day-to-day world, I am continually writing down mathematical models, testing the data, usually discovering that my model is incorrect, and then going back and trying again. I do think that it's very important in this kind of work to try and do formal testing of your models. And that requires some sort of mathematical knowledge. It's worth remembering, though, that the model is just a model. There's a famous quote in statistics, not from me, that all models are wrong, but some are useful. And that's really the point here. Of course, the true complexity is such that we could never hope to write it down. Even in ancient populations, people are moving around, new immigrants come in who may be genetically rather unusual compared with where they're living, and so on. And so actually, it's very important to get a reasonable number of samples from a population. Many of you will have heard of the famous Iceman, who was recovered some years ago on an alpine glacier. And the people who discovered him saw this body on the glacier and thought there'd been a murder, and they were right, but the murder took place in 3000 BC. And we looked at the genetics of that and discovered that this unfortunate individual had genetics that looked quite like modern Sardinians, and nothing like the genetics of the people who are living in the Alps today. So what does that mean? Well, I was very cautious when that first came out, because, okay, he looks like modern Sardinians, but maybe his mother and father had come from ancient Sardinia and moved into the region, and he wasn't at all typical of other people living there. That now turns out not to be true. We've now looked at a whole number of samples from 3000 BC Alpine area, and in fact, he seems genetically fairly typical of those people, and they did indeed look like modern Sardinians. And the reason for that turns out to be that modern Sardinians are the only modern population that look quite like the first farmers in Europe. And this Iceman died before the steppe people had arrived, so he looks like a farmer.